My name is Jacqueline Williams, and I would like to share the research I conducted with Dr. Tom Delia and Chris Holland at Indian River State College in collaboration with Dr. Blake Ueshima, who was at the Smithsonian Marine Station during the research and is now at UNC Wilmington. The title of the project is Isolation of Vibrio Coralliilyticus Bacteriophages from Florida Seawater. Vibrio Coralliilyticus is a gram-negative proteobacteria that is an important coral pathogen. Infections typically result in destruction of host tissue, while some experience bleaching, which may result in complete coral colony mortality. This bacterium has been implicated in various outbreaks of diseases, collectively termed white syndrome. The significance of these infections is the rapid decline of coral health negatively impacts many marine industries, such as tourism, fishing charters, and seafood consumerism. Currently, there are only a few options for complete control and prevention of these diseases. My objective was to isolate and characterize new bacteriophages from Florida seawater and sediments that are able to infect V. coralliilyticus with the intent of screening them for the ability to be used as phage therapy as part of treatment against V. coralliilyticus coral disease. Bacteriophages were isolated through enrichment protocol and purified to achieve purified bacteriophage. In Table 1, there is a list of all the V. coralliilyticus strains used in the study. The first one listed is OFT 621. This is the strain that was used for the original isolation. The remaining strains were used later for host range analysis. The strains represent bacteria that were collected from different geographical areas to account for possible genetic or biological diversity. Figure 4 shows the purified plaques for 3 isolated bacteriophages, phages 828, 99, and FLO1. Phages 828 and 99 were isolated from the samples collected from the Fort Pierce Inlet, and FLO1 was isolated from the samples from Birthday Beach in the Florida Keys. All isolated phages produced clear plaques indicating lytic life cycles. Lytic bacteriophage are important in phage therapy as they do not have the dormant lysogenic cycle and ensure killing of the pathogenic bacteria. We completed electron microscopy of the phages 828 and 99 at the USDA in Fort Pierce with the help of Dr. Wayne Hunter. The phage morphology is consistent with myoviridae, and the images show that the phage tail sheaths are extended on the left and contracted on the right in the EM pictures. Next, we tested the isolated phages for their ability to infect the different V. coralliilyticus strains to evaluate how wide a host range the phage might have, an indicator of the phage's ability to kill the different strains that might be present in natural coral infections. Table 2 shows the host range assay results. Phages 828 and 99 are capable of infecting host strains collected in Fort Lauderdale, but not the Florida Keys. OCN 14 was the only Pacific Ocean strain that phages 828 and 99 could also infect. Figure 6 shows the positive results of which strains the phage were able to infect with varying efficiency based on the titers produced on each plate. In conclusion, there were 3 Vibrio coralliilyticus bacteriophages that were isolated from seawater and sediment, 2 from the Fort Pierce Inlet, and 1 from Birthday Beach in the Florida Keys. All phage produce clear plaques suggesting lytic life cycles. The lytic phage are required for successful phage therapy applications. The phage isolated all show a myoviridae morphology, consistent with the V. coralliilyticus phages previously characterized. The phages show the ability to infect more than one strain, and more importantly, they could infect strains from Florida. This result suggests that the unique phage for phage therapy we need to be purified from the location of the disease to increase the likelihood of the phage being able to infect the V. coralliilyticus from that region. We are continuing this work and in the process of sequencing the genome. We are also hoping to conduct phage therapy trials where phage would be added to diseased coral alone and in conjunction with current treatment and evaluate the impact on the disease progression. Thank you for your interest in my project, and I look forward to discussing any questions with you.